Hi everyone, welcome to my channel. Today we will discuss about DNA and RNA and why DNA is a genetic material and RNA is not, even though both are nucleic acid. So what is nucleic acid? Nucleic acid are polymer of nucleotide. That means many nucleotides are bound together to form nucleic acid. Also essential for content are genetic information. It is a biomolecule, by the way. See, this is a nucleotide and another nucleotides are bound together to form nucleic acid. We have majorly two types of nucleic acid. One is DNA and other one is RNA. What is the difference between them? DNA is double stranded and RNA is single stranded. DNA is helical structure. So we have a pentose sugar. It can be ribose or deoxyribose. Means it can be RNA or DNA. So first carbon of ribose sugar is attached with nitrogenous base, and fifth carbon of pentose sugar is attached to phosphate group. The bonding between first carbon of pentose sugar and nitrogenous base is N-glycosidic bond, and the bonding between fifth carbon of pentose sugar and phosphate group it called phosphoester bond. Okay, so you will ask what is phosphodiester linkage. The phosphodiester linkage is nothing, two nucleotides are joined together, phosphodiester linkage. Okay, two nucleotides are joined together with phosphodiester linkage. Okay, an important thing, phosphate group and sugar make backbone of nucleic acid, and nitrogenous base is projected inside at 90 degree. Okay, we have DNA and RNA. What is the difference between them? See, two carbon of ribose sugar has hydroxyl group, means OH group in RNA, but in DNA only hydrogen. This small oxygen atom create lots of difference between structure of DNA and RNA and their mutation rate and their stability also. Now we will discuss how it affect their stability and mutation, because the RNA contain one extra oxygen. It is very reactive. That means RNA is very reactive, and DNA has only hydrogen, not an extra oxygen group. That means it is a less reactive. There is also difference between their bases in DNA and RNA. DNA and RNA almost have same basis except one. DNA has thymine and RNA has uracil, which creates lots of difference again in their mutation rate, their stability also, because uracil again is highly reactive in RNA. So RNA again is highly reactive than DNA. Criteria to be a genetic material, it can self-replicate itself. So RNA and DNA both can replicate themselves. It means they can make copy of itself. Second, chemically stable. So I already said to you that DNA is highly stable because it has lack of one extra oxygen atom, and RNA is highly unstable because it has an extra oxygen atom. Mutation rate. Mutation is important for evolution, but too many mutation can be harmful. So DNA mutates slowly, but RNA mutate fastly because RNA has an extra oxygen atom which is very reactive. That's why RNA mutate fastly. Fourth, it can express itself in Mendelian character. So again DNA can express itself in Mendelian character. Let's discuss who fulfill all the criteria to be a genetic material. So DNA fulfill all the requirements to be a genetic material. It can replicate itself, it is chemically stable, it mutates slowly, and it also expresses itself in Mendelian character, which RNA does not. RNA only explicate itself, otherwise it not fulfill all other criteria. So DNA fulfill all the criteria, so hence prove DNA is a better genetic material. So what is the function of RNA? Why RNA is exist if RNA is not express itself? No, that is not completely right, because scientists propose RNA world hypothesis. This said that RNA is the one first genetic material on the earth, which is also present in most of the member of Monera kingdom. It later evolved itself in DNA, because DNA is highly stable and it can contain genetic information for long time. So DNA is better genetic material. But otherwise, RNA also contain genetic information. It behave like a catalyst. And RNA has majorly three types: mRNA, messenger RNA, tRNA, transfer RNA, rRNA, ribosomal RNA. These three type of RNA can synthesis protein. So RNA is also important for protein synthesis. Let's discuss the difference between DNA and RNA again. So sugar type, DNA has two deoxyribose sugar and RNA has ribose sugar. Nitrogenous bases, DNA has adenine, thymine, guanine, cytosine. RNA has uracil instead of thymine. One more thing, adenine always hydrogen bond with thymine, and guanine always bond with cytosine. In case of RNA, adenine bonded with uracil. Okay, the structure. DNA, most of the organism is double stranded, but RNA is single stranded. In case of retrovirus, it is a double stranded. Chemically stable, DNA is highly stable and RNA is less stable, most reactive. Mutation rate, DNA mutates slowly but RNA mutates fastly. Primary function. What is the primary function of DNA? DNA is a primary function is storage of genetic information, but RNA functions are different. It can be catalyst, it can be helpful for synthesis protein, it can be genetic material. We again discuss the central dogma, which is proposed by Francis Crick. He said that DNA only expresses itself when it converts into an RNA. Otherwise, DNA cannot be expressed itself directly. DNA always convert itself into RNA, then RNA make protein, which express character. So DNA convert into RNA. This process is called transcription, and RNA convert itself in protein. This process is called translation. And lastly, thank you for your attention. If you like this explanation helpful, please like and subscribe. We will meet in the next video. Bye bye.